But because of my parents' background, I grew up learning a lot about hard work and discipline, and doing things like watching my father build the cabinets in our house, but also learning that I was in deep trouble if I didn't remember to close the cabinet door. So there was a good amount of discipline in our life. My father was a part of the team of nuclear engineers who started up the Indian Point power plant in New York in the late 50s, early 60s. And we all know now that that power plant is about to be closed down. He would study on his own late at night to train himself to do this. He worked for Consolidated Edison, and when you got a job at a place like Con Edison in those days, the idea was that that job was for life. Today's world is completely different. You don't ever think, and should not think, and this is one of my big lessons, that a job for life is not the route you have to take. But I grew up in that head that you get a job for a big company and you pursue that career in that company forever. And what is also true is I'm a gal talking about that. So my mother had a career in the phone company, and it was just understood that I would go to not only college, but graduate school and pursue a career. So I hope that gives enough of a flavor.
Yep. So you ended up going to Connecticut College and studying botany. How did that happen, and why botany?
And so how fun for me, because recently the botany department asked me to come back and answer that same question. My mom was an avid gardener and knowledgeable, not just liking to pull weeds, but she knew about plants and botany and horticulture. And I learned that the fun of being outside. But the reason I studied botany at Connecticut College was because at that time, again, this is 1970, just to put a date on it, the college was one of the only places that was doing ecology and bioecology. And it was the place where a movement started to preserve and understand the importance of protecting salt marshes. And the salt marshes of Connecticut, by government proclamation, were protected and conserved early. And we investigated and did lots of dig in the mud, plant and animal analysis of the bioecology of the salt marshes in Connecticut and Rhode Island, and that was really cool. Yeah. So why did I do that? I did it because I loved the team of the department. It was a very small department, and they were very passionate about what they did, and we all were very connected with the vision they had, and that's really why I did it. Yeah.
So you then went on to get your PhD at Harvard and studied molecular genetics?
Years later, but after college, I didn't know which end was up because I didn't really think I was going to get a job teaching botany or working at the Brooklyn Botanic Gardens because remember I told you that the career path was a job in a company. And so I spent 4 years, 2 years of them working at Cold Spring Harbor Labs on Long Island, which is where Jim Watson came after he did the work on determining the structure of DNA and getting his Nobel Prize. He ran Cold Spring Harbor Labs, so it was really a hub and a bustling place. And that is where I learned and had the fun of not only do you get passionate about doing science and running experiments, but you get passionate about working with really smart people and then going out and having beer and then coming back and working even more. And I also realized that I didn't want to live on Long Island for the rest of my life, and I moved to Boston and did the same thing exactly at MIT, at which point I got kicked in the behind and told it was time to go to graduate school. And so my MIT mentor, Harvey Lodish, went along and got me going to go to graduate school. Thank you. This is luxurious, and if you're saying like, does someone just like raise your hand if this is like dull?
If anybody feels gutsy enough, I guess they're welcome to.
No, it's more fun if people.
But yeah, if anybody has questions, feel free to ask a question. But don't ask like 7 questions in a row, because it will never get to the actual Q&A portion. So did you know going into grad school that you'd then leave to go to industry, or were you thinking about a career in academia?
Well, my graduate school was with this guy, John Beckwith, who we were working on bacterial systems, regulatory systems. He was the guy who figured out what the hell an operon was, which is a fundamental unit of regulation. We studied bacteria, but it turns out it's the same in humans. But John also was a social activist and wrote copiously articles about the lack of a genetic correlation between women's ability to do math successful and be successful. He also didn't believe that there were genetic determinants for criminality, and he did and wrote about that. So we spent a lot of time, because we had a collaboration with a laboratory in Paris, going out, having Calvados and beer, talking about social matters as well as science. John thought industry was corrupt, because now, let's just think of the date, let's do a date test, we're in 1978. And it wasn't customary for PhDs from Harvard to go off into industry. So this was a turning point for me, clearly. John is a guy who gave one of his awards from the Eli Lilly pharmaceutical company for recognition of all of his science to the Black Panthers for their trial. This was certainly not appreciated by many of his colleagues at the time, but that showed his commitment to social issues. So no, my choosing to decide to go to Pfizer was not something that he supported or ever understood. But many of his students subsequently have chosen the path, so I guess he's lived with it. I guess the other learning was that John did teach us about the importance of writing well and communicating well in science and articulating big thoughts simply. And I really admired him for that. And he also is the one who kicked me in the other side of my behind to do a postdoc in a different field, because we were a club and we all really enjoyed working in these bacterial systems using genetic tools. But I was driven to study cell biology, and I came to Yale and went to the cell biology department. So I began to think about bigger systems and really learned that I'm much more of a generalist in science and didn't drill down as much. And that enabled me probably to feel very comfortable interviewing not just for traditional academic jobs when I was doing my search for jobs, which I did do. I interviewed for traditional jobs with a research plan in hand in my slide deck. But then I also interviewed at Pfizer. And when I interviewed at Pfizer, which was down the road in Groton, I just knew that the idea of connecting basic research to drug discovery and working with the team that I met during my interview would be the best thing since sliced bread. And it really was. It was really very cool. And so now we're at 1985. And I stayed there at Pfizer for 10 or 11 years, half the time running a lab that did drug discovery, small molecule drug discovery. And I had lab assistants and was part of a larger team. I was the biology arm of another team that was the chemistry group. And it was very fun to be part of Pfizer at that time. It was a huge, great, cool organization. And what you learn to appreciate is the incredible depth of knowledge and talent that a place like that has, and resource that you can draw on.
So at some point you made a transition to licensing. How did that happen?
So I was talking to someone about this earlier. I learned that the biggest, best fun is the science-business interface. And I did it when I was running my research lab at Pfizer, because I put a collaboration together between my lab at Pfizer and this guy, George Weinstock. Some of you might have heard of George. He's up at Jackson Labs now, 100 years later, doing microbiome research. And I loved that interaction with George. He was at the University of Texas at that time. And when a job came up in the business development group, which was not really just licensing, it was collaborations. I snapped at it, and it was really fun because I got to put very big deals together with a team of Pfizer scientists. In particular, my portfolio had a lot of tech deals with a bunch of, well, I call them tech. At the time, new technology for doing drug discovery, like new methods for doing chemistry, fast chemistry, scale-up chemistry, different kinds of chemistry, new technology for building detection systems for measuring fluorescence in 360 well microtiter plates. So I did a suite of deals called the Big Four, and we closed all the deals in one day, and they were $200 million of investment. So what you had to do was get the Pfizer scientists on board, get management on board, get the companies that you're doing the deals with on board, sell the whole package to the senior manager, to the CEO of Pfizer in pitches, and then put the legal agreements in place, manage the IP, build the teams that were going to manage each of these collaborations, because every collaboration at that level, we met with the company once a quarter. And so that was 4 times a year with 3, well, there were 4 deals, including a company in Brantford that no longer exists, Neurogen. So that was fun. But at the time, I figured, well, where do I go next? And even though by that time my father wasn't around anymore, so he couldn't scold me, I said, I don't want to work at a big company for the rest of my life. So I left and started my own biotech, because I knew I had all the tools. I knew how to build teams. I knew how to manage intellectual property. I knew how to put legal agreements together. I knew how to pitch. And I had the science background to put the story together.
What was the seed or the catalyst to actually make you take the leap?
I just got bored with doing the same thing.
Mm-hmm.
I still love the people. I still valued everything about the company. The culture at these big companies, though, changes over time, and at that very particular time was when Pfizer was beginning to start its round of many, many mergers. So that was the beginning of all the many, many internal meetings to plan for change. And over the years, I missed out on all of them, and they still happen because they're still changing in these big companies. They manage change. And at that time, they were, at Pfizer, they were very, very, very careful about managing to change because they didn't want to disrupt the momentum and the productivity of people. Now they're less careful. They just take you to your office and say, I'm sorry, you're not going to be here anymore. Just take your things. They didn't do that at the time. So it was a very transitional time, but that wasn't my particular driver. I wanted to do something else. Plus, I had a friend who's one of my co-founders, and we had many, many bottles of wine in which he said, so Suze, when are you going to start the company? When are you going to start the company? So that was the driver too.
What did your team look like when you started?
Me, It was brief. I worked for quite some time at home thinking about it, figuring out what I needed to do, what the moving parts were. And my science co-founders who just said, okay, when are you going to start raising some money? When are you going to start? When are you going to start? And finally, we had a huge meeting of the three co-founding scientists who are here at Yale still, and although one guy is an emeritus now, and a gentleman who had founded and made tons of money on a biotech in California, who's a dear friend, and another gentleman who's around this biotech community here in Connecticut a lot. His name is Harry Penner. He's done a lot of startups, and I knew Harry from deals I did with his company, which was Neurogen at the time, in Brantford. And Harry joined this powwow or meeting that we had at Tom's house. It's Tom Stites, who's the main co-founder. He went on to get the Nobel Prize for his science for the crystal structure of this molecular tool called the ribosome, which was the basis of the science of the company. And we this group of co-founders, just said, okay, it's time to raise money. Why don't we just all put in some money? So we all made a decision to write checks, and then we asked some of our friends. And so we had we put a bridge loan together, which was a lot, but that we were all older and able to have that luxury of doing that. And that's what a lot of biotechs don't have, and that's why it's hard sometimes to get going.
Do you find
it was $2 million.
As when you're doing advising, when you're exposed to other biotech startups, do you find people writing their own checks is particularly helpful? Does it get more buy-in, or is that really independent from the likelihood of success?
I think investors are quite taken with the co-founders having some skinny in the game early on. There's certainly acknowledgement that young entrepreneurs who are just starting out in their careers don't have the capacity to do that. So so sweat equity is certainly expected and considered very, very important.
So when you launched, you had some sense of what the science was. Did you know what the practical application or the monetization with the end product was going to be, or were you saying, look, we have some really interesting science, we know we can do something with it?
Well, so the part of the story that I didn't mention is that what I did at Pfizer was, and because my background from my PhD was in microbiology, I did antibiotic drug discovery at Pfizer.
Mm-hmm.
And so the company was about new antibiotics. And since we understood very, very much, the scientists and I, very, very much what this asset was that we brought in from Yale, from Tom Stites and Peter Moore, we absolutely understood what the goal was. But we understood the goal in a very, very general way. The business model of the company did not change for the entire 11 years, or for probably even now, the 17 years that the company's existed. The idea being to use chemistry, molecular design to synthesize the next generation of antibiotics, and to do that iteratively, to build new molecules, new chemicals, small molecules, like for pills and bottles, that target these very serious drug-resistant bacteria, which you are reading about again, and that there's a problem. So we knew that, but did we actually know what the product description would be of the first product? We thought we knew it would be against MRSA, but I will drug-resistant Staph aureus, which was the gram-positive infection of concern of that time. But it's 17 years later, and they just launched their first product. And so it was positioned differently than just MRSA. And so there were many, many complicated turns and changes in where the chemistry and the medical need and the buyers and the investors were going. And those are things that you can anticipate in a perfect world, but I think drug discovery is not moving from A to Z. It's science, after all. It's research. And research, if anyone who does bench work realizes that you can't just demand that science deliver what you want it to.
Yeah. So how did you get through the times when science didn't deliver what you wanted to?
And that would happen almost every other day. It's very, very stressful. I mean, the main job, of course, was raising money because drug discovery is hugely expensive. So our first Series A was pretty darn good. And I don't think you see it that much except for just a few companies now in Connecticut. You see it for Biohaven. You see it for Craig Crew's company, Arvinas, where we raised first round $50 million. And so we hired 60 people, and we built labs in George Street here, and we started cranking. And so, but you run through that cash very quickly, and so we're constantly having to deliver results and go out and raise more money. But at the same time, you're building your milestones and your deliverables, and you're having your board meetings, and you're going to promise the board these data, and the day before the board meeting, you get those data. And sometimes we didn't even understand why we got those data for the board meeting, and we'd have to, come up with the story. I mean, the neat thing about the company, and this is not for nothing. I mean, Erin Duffy still leads the science there. She's the chief science officer, and I hired her right out of the bat. So she's been doing it for 17 years. There's the integrity of the people we hired and their commitment to the excellence in their research, and the mission was unbelievable. And that they knew science and their particular expertise way better than I ever could is also remarkable. And so the great fun of this was the unbelievable superbness of the team, the humor that the team was able to have, but the credibility in front of investors and at the board meeting when the data were so not what they were supposed to be. And it would be from not being able to make the bloody thing at the scale. I'm using some jargon. You have a small molecule, a chemical, and it's great if you have like a speck of it so that you can test and ask if you can kill bugs, bacteria. But you have to scale it up, and you have to be able to put it in a mouse, and you have to be able to put it in a rat to make sure you're not killing rats with toxicity. And you have to be able to do that again. And if you can't make more than a little sparkle of the molecule, you're cooked. And sometimes you just couldn't. Or sometimes it would take 2 months. And so those kinds of hurdles slowed everything down and certainly caused a lot of angst. So the CEO's job is really unpleasant because not only are you managing all these people who are essentially very independent because they're so smart, and they are not accustomed, scientists are not accustomed naturally to work in teams, not naturally. And you have to manage the investors, and you have to manage to an ever-changing molten budget, because if you have to redo something, your $100,000 investment in a particular study, I'm using rats because they're kind of a little more expensive to work with, but you have to make sure you're not killing them. It's a safety test. It costs a lot of money, and so a budget shift of $100,000 can happen daily. And if you think about it, well, that's an FTE, almost a person, a full-time, or it's the purchase of a piece of equipment, or it's paying the rent, or it's quite a lot of things that you have to weigh. And I'm using a simple example in order to even just redo something that just screws up. And that happened all the time, and even at the level of the clinic. And that's real money when you're paying for studies in humans.
How do you stay sane when stuff like that is happening all the time?
My disposition was not always cheerful, I will assure you. And the worst of it was, it's hard to unwind. You think of it, thinking it and breathing it and living it 24/7. And that means you give up other good stuff if you're not disciplined. And you give up a certain quality of life on the home front. Because your partner, if you have one, if you're lucky enough to have one, who's putting up with all this, gets really tired of, well, so-and-so did so-and-so, and that investor, he just won't answer my phone call, and what do you think about these data? I mean, it's hard, and that's all it is, and that's why I recently quit, and I went to Tanzania for a month. How cool is that? Yeah, so.
That sounds pretty nice.
Yeah.
So when you're hiring, what do you look for in an employee, and in teams do you look for a collection of different skills or personalities?
Yeah, so I'm not sure I did it well, except to say that the initial structure group and some of the core chemists are still at the company 17 years later, so that's good. But we just looked for the best people, and that meant that not necessarily always were the people the best team people, because sometimes it doesn't go hand in hand that you're a great team player if you have a lot of independent thought and skills. That's the truth.
To that, how did you know, being a scientist, that you would be able to be a team player and then be a team leader? How did that develop? Because you just mentioned that the personalities sometimes don't work in teams.
I don't know how great a team leader I was. I mean, I have a style. I'm very direct. I'm very transparent. It wasn't always a democracy. You have to be brave, and you have to be willing to tolerate tons and tons of risk. And you have to tolerate people not liking you a lot all the time, which isn't so great. I hired a lot of people who I knew and connected with from my days at Pfizer, and who my co-founders knew, and that helped. And so it was a lot of word of mouth networking that allowed us to build the early core team. And we modeled our comportment as a culture early on. We had a couple of days with a psychologist who loved organizational management, and we modeled a lot of it on similar trainings that the Pfizer people among us had had. So we loaded it up with some of the Pfizer principles of team listening skills, really quality listening, just certain methodology for being courteous, and whatnot. But we also hired an HR person because even in a little company, recognizing that issues really come up very early, such as harassment issues and other things, and we dealt with them. even early on in a company. And you have to be motivating for your employees. You want them to be passionate. You know, it's one thing for the senior people to be there, but you want everyone. And you have to build an organizational structure. People need to know where they sit in an organization, what their hope is for being part of things. And a little company can be part of a lot, and a leader needs to understand that and figure out how to do that. And you have to have events. You have to have parties and all that stuff. And that, I think that helped the team building part, because teams don't automatically gel. So like, be a team.
So I might be jumping ahead a little bit, but you,
late, isn't it? What time is it?
make sure we
Is it
don't
8
go.
o'clock, 10 o'clock?
Not quite.
Okay. The fire doesn't burn low. What
No.
goes on with this fire? It's supposed to like settle.
It's amazing. You spent some time at UConn in technology exchange, and you're involved with Yale's OCR. What have you seen in commercialization of technology that works, or signs that something isn't going to work?
Well, just because a faculty person, wherever, chooses that his or her research should be commercialized, doesn't mean that it is commercializable. And that's managing those expectations is one thing. And you're asking me, well, what is commercializable? You have to, in my case, understand that there's a medical need, that there's actually is a market, that someone, that an investor will invest in it, and that a partner, such as a big company that can write a check to help you do clinical trials, has a portfolio in which your idea of a product fits. I mean, we had an idea of a product for MRSA, and so there was high medical need at the time, and so that was easy to attract investors to. So commercial, you have to do that business assessment. What also doesn't work in my experience is, and I probably get hit with a brick, is that faculty, just because they invent something, doesn't mean that they can run a company or put their 50 cents in the middle of a company. And when I have seen that happen, where there's been that expectation that it's the faculty person's company and that individual is more than a scientific advisor or an expert advisor, I've seen that does just not work.
Yep.
My founders had wanted nothing to do with the company except to advise on the science. So they helped raise money because they had branding. That's the truth. They were branded with their Yale personas and their accomplishments. But after that, they didn't want part of it, and it would have been very complicated if they had wanted to be part of it. But they also had ethical reasons because of NIH guidelines and Howard Hughes guidelines that they didn't want to be that involved. What didn't I answer?
I think you answered the question.
Okay, and what else doesn't work in tech transfer? I think it's a tough exercise to figure out in a university portfolio of faculty accomplishments what should be patented and what shouldn't, because everything can't be. It's just too utterly expensive. And so the job of a tech transfer office is really hard because they don't have an infinite budget, and they have to make really good guesses on, not guesses, informed, advised decisions about what to commercialize or what to patent, I think is really the fundamental question.
I could ask a lot more questions, but then we'll never get to Q&A, so I'm going to go ahead and open up to Q&A. But if nobody raises their hand, then I will.
I would love to know if you pick a protege. I mean, we always run into problems in companies of things not going like we promised. So what advice would you give to that protege when you're first going back to the investors to ask for more money, but things are not going as planned?
well, you can't surprise the investors, right? They need to be on your team. You need to not just go off and have a disaster and then tell them 6 months later. So they need to be in the room, and they need to buy into what the pivot is, or they need to have a deep understanding. One wants investors who buy in and are not casual, and that also means, especially in the case of biotech, that they get it. They get that it takes a bazillion years and literally a bazillion dollars to get a pill in a bottle. And they need to understand where they fit into that timeline, how much they can invest, and express that commitment. Because if they run away every time something icky happens, you're cooked. So you have to choose your investors, carefully.
During your time in Tanzania, right? You said you just escaped?
Yes, I got this.
Did you have any sort of realizations once you got out, right, and reflexively thinking about your time there? Was there anything that sort of the circumstance of escaping helped you put two pieces together?
So it happened before Tanzania, because I started this exercise of transposition. I went to Botswana and Patagonia. And thank you for asking, because it brings back this fundamental love of the natural world that I figured out I had from my botany days. And for crying out loud, if all you do is spend your time in your office talking to investors or at team meetings, this entire world goes by, and you miss it. And I learned that my ability to focus incredibly on every animal study in 100 molecules for a month, a day after day, I can use that same focus and intensity in sitting and watching how a bird behaves in the wild, and just absorbing it and being amazed at the diversity of nature. And so it allows you to just absorb the world, to be really remote. I mean, these are going off and being remote, not going off and being in Marriott hotels in the desert. It is very, very cool, and I'm very lucky because I get to go. There are 4 of us who've done this now for a couple of years, and we all share the same passion for scientists who go off and do it. It is really, really fun, and I would recommend prioritizing and figuring out a way to do it, because it's not about money, it's about time. And in this particular case, I'll go on just briefly to share it because it was cool, is seeing the Maasai culture, the Maasai warriors who still exist, and the adherence to the ancient Maasai ways that still exist in the northeastern Tanzania, which is called Maasai land. And it is remarkable. It is interesting. And it's harsh. A lot of people live pretty harshly compared to us. Is that enough? Any other questions?
Any more questions?
It's super cliche, but if you had to do it over, how would you have gone and done things differently and starting?
Faster. I wouldn't have messed around so much.
What do you mean messing around?
I had too much fun. I was having a lot of fun when I was working at Cold Spring Harbor and at MIT, because I was carefree. I was just, going to work, hanging out, doing experiments as part of it, but I had no responsibility. And it was just fun. But it's also, graduate school was fun too, but I was sort of more directional. And I had spent too long in graduate school. I didn't want to leave there either. I was there for 7 years. That's a long time. So once I started figuring out what I wanted to do, this idea of working in the science-business interface And this notion of being an entrepreneur, where does that come up? How do you figure that out? How do you put a finger on that? No, I had no community of entrepreneurs to talk to. That goes back to your early question. Certainly, I mean, you're entrepreneurial when you're doing your PhD thesis and you're trying to salvage your thesis and figure out, well, those experiments aren't going to work. How do these work? But it's not quite the same as what you all are engaged with. And you have a place to come and network and meet people and connect. But that didn't exist. That was unheard of when I was figuring out my company. So now you know how you sort of have when you asked who helped you, and I said it was me. Well, it really was just me, and that's lonely.
Two things. One, I think you say you would change it and make it faster, but it does seem that you ended up diving really deep into your botany in a lot of ways. Like if you look back at what you just said and how you rediscovered that part of the ecology, really what you've done over the years is gotten even deeper into what makes the world and how you change those things to alter the outside of that. So I think that for me, like listening to your story, it seems really interesting how, though it may have been a divergent path at times, you still had to focus to get to where you want to get to. And two, as an entrepreneur, what are some things that you've done in your life or now that you would suggest to us in the same world to manage stress and manage the world around us? Because that's one of the hardest parts, right?
Yeah, it is the hardest part. Now there are more tools. I think there's a yoga center, for example, around the corner. I think finding a group of friends who you can share, friends, not family, friends who you can really share. Now there are CEO networks for crying out loud. You can go out and and one of the CEO networks is meeting right now, of bioscience guys, and have a beer and share, and that really helps too. And then you just sort of get out of your system pretty fast. I think making sure you have a mentor who you can trust, really, really trust, and who you really respect, and probably someone who's more senior and who is very accomplished in your court is very reassuring, because everyone gets unsteady from time. Everyone is unsteady from time to time, and so you have that person. So you're not relying on your family to buffer all that, because it's a lot to ask. It makes sense.
Yeah, that makes sense to me. Sounds good. Any more questions?
So, just going back to the beginning of Rib-X, you mentioned Tom Stites and Peter Moore. The question is, how did you find your other co-founders, like Harry Penner, for example, and others?
Yeah.
And what were the steps? How did you get your first dollar in the door? That was
I can't quite. How did I get the first what in the door?
Dollar.
Oh, well, I mentioned that we all put in money as co-founders, and we asked a network of pretty well-established friends to do that. So we got, in our bridge loan, I mentioned it briefly, but it's a little embarrassing. It was $1.8 million. We cut it off. That's a lot of money to go around with in the bank to use as in hand to talk to investors. But that's how we did it. That's how we got it started, okay? The other co-founder is Tom Stites and Peter Moore, who was the original science, and then we needed another, we wanted to do computational chemistry, and I knew Bill Jorgensen, who's in the chemistry department here, as a collaborator from Pfizer. And so I brought him into the group, even though he was a colleague of Peter Moore's. They hadn't really collaborated much, so they brought him in. And then Harry Penner, I knew because I had done deals. I put deals together when I was at Pfizer with his company then. His company then was in Brantford. And I did a bunch of Pfizer deals between Pfizer and his company. So I got to know him then. So that was lucky, right? I used my network. And then there was one other co-founder, so that was Tom. It's Tom's friend. We brought him in. So that was it's an unusual story. But when you hear like of companies like Biohaven now, Vlad, he probably has a very similar kind of maybe shifted story, you know, high net worth individuals and experienced people that he was able to put together. But that's not common. And then Craig Crew's story, his company is Arvinas. That's another company that raised tons of money in the beginning. Is here in New Haven. Craig's a faculty member here. He doesn't have tons of money. Well, maybe he does from his first company. But he had success. He'd done it before. He was a serial entrepreneur and really, really brought in money for his investors, the first company. I can't leave without saying, though, that networks are really important. I mean, the buddies I made having so much fun at Cold Spring Harbor, I still can connect to and bring in. And we're all old now, right? So most of them are all in interesting places, and so you can draw on their connections and their. And so as you are young, it's important to build your network because you never know.
All right, we've got time for one more question. Back.
It seems like you, across your life, you've either had in you or developed a very diverse skill set to be successful in what you're doing. And so what I'm curious about is, what was your process for saying, okay, here's a skill set that I need to learn better, managing my employees' morale or something, or legal contracts. How did you say to yourself, gee, I need to learn this, and once you sort of figured out that, how did you go about learning that skill set that you might have not had to start with?
So I hired good people to work with me. I didn't have to learn all this. That is the honest art of building teams. You need people who are way better than you are and more knowledgeable than you are in all sorts of things. My skill set, I think, is, I started to say it before, something that who knew at the time, is being entrepreneurial and willing to live with risk, living with that adrenaline, that constant adrenaline of risk. And you know, you have to be a little smart. I mean, you can't be a dummy, but you have to not want perfection. I mean, some of the scientists I hired wanted perfection in everything that they were really, really good at, and that's why they were successful. But they could never have been the CEO, because if you want to live in the world of perfection, you're never going to, move fast, which is one of the things I said that I think is important to try to do. Move fast. I think that makes sense.
Yeah.
I didn't go off to the library and study patent law, although you laugh at me. I did take a patent law course. But I wasn't a good student of it. I learned enough. In other words, I didn't take the exam. I didn't become an examiner. Anyone can become a patent examiner. You don't have to, you just have to take the course. It's true. I'm not making it up.
Thank you so much for joining us. Thank you.